So we used to have MIDI imaging here in the BayBioMS. Unfortunately, our MIDI just died. So the laser broke, and it is too expensive to repair it. And also, I will come to the end. It is a nice machine. Michael Schrader also likes it. But doing MIDI imaging, it's much more fun if you have a high resolution mass spec, and we still have those machines around. So that's why I think it's still interesting for you to know this technique and see if this might applicable for your research. So when we started off, we did imaging of carrots and potatoes because I'm a food chemist, and I will basically walk you through the applications. So what is the benefit of mass spectrometric imaging? You get a spatial distribution of specific metabolites, lipids, peptides, or protein within a tissue. So normally this is done for tissue. And for example, what people are trying to do more and more is like typing of tumor tissue. So if you go into like the pathology department or have a biopsy, usually like the manpower looks at the biopsy and then decides this is tumor tissue or not. And if you try to move this technology to a mass spec, which can also say tumor tissue, non-tumor tissue, it can also work day and night, 24/7, and it don't have just 8 hours per day to work. So that's why there are huge applications moving into tumor tissue. Then we have drug delivery, and I think this will Nicole most likely talk about. We can use it, for example, to identify biomarkers and stress markers, and basically it might help you to get a better understanding for biology due to the spatial resolution. And so what has been done so far, I'm not sure if somebody has joined so many seminars that they still remember it. We had a student from the BGT, the Brewing and Beverage Technology Chair, who did MIDI imaging of wheat and like to see the process of malting. Then we had here a bachelor student who basically, when we got this technique, developed the method. She's now a postdoc with us, and we did carrots, potatoes, and stuff like that because, you know, food-related field. And then we had this was published last year, a PhD student who did MIDI imaging of white adipose tissue of mice. Unfortunately, he already left, otherwise he would have been a good fit today to present his data as well. So basically how the MIDI imaging is separated. So we need to prepare a thin slice of our tissue, plant, whatever you want to put into the machine. Then we need to apply matrix to it. Then we do the measurement, and then afterwards you need to try to make sense out of your data. So I will quickly walk you through all those for type hikes. So basically, we use a cryotome. This is like the instrument people use. I'm not sure if you're do H&E staining. This is like a common instrument used for preparing those slices. The difference is that we always need to put our desired object in some kind of matrix. And what the H&I, as normal histological staining people use, is OGT. This is like a really excellent source for MS detection. So this is not a good choice to put your tissue in for MIDI or mass spectrometric imaging. So that's why we use, for example, carboxymethylcellulose, agarose, something which is quite inert, are not detected in mass spec. And you need this matrix so that the cuttings are getting easier. And like to optimize this cutting, you think it's quite easy, but it's not. It takes some time until you have the perfect cut. And then you need to adjust the temperatures if you have high fat, or if you have like a more porous compound. And sometimes the student from the BGT, she used even TESA to get her wheat samples onto the slide. And then basically you are limited to the normal glass sides you all know. The only difference for MIDI imaging, you need to have a conductive glass slide. So one side of this glass is conductive, and then add access. So later on, you need to teach the mass spec where your tissue is, what you want to image. And then I will talk about why you need the matrix in the next slide. Nevertheless, you need to add matrix to your sample, and then there are different options. What they all have in common is that they have an aromatic structure. And then there are like rules for peptides, HCCA works best, for proteins, DHV or SA, cyanophilic acid. And then when you start off your multi-imaging project, you just, try one or two and see where do you get the best results. And we have this image prep station here, which is like spraying the matrix onto the slide so that you have a really equal distribution of the matrix on your glass slide. And you need the matrix because we do matrix-assisted laser desorption ionization. That means the laser shoots on your target. You have your sample molecules and the matrix, and basically the laser excitates the matrix, and then there is happening the desorption protonation process so that the charge is going from the basically matrix to your molecule of interest, and we can only detect charged molecules with our mass spectrometers. So sorry. And then our charged molecules go into the TOF, so it stands for time of flight. And the longer the TOF tube is, the better is the resolution. And in the case of our instrument, which we had here, we had a reflector mode. So here's basically a mirror, which mirrors the ion. So you have a longer drift tube, and with that you have a better resolution. So then you have your mass spec, you have the matrix applied, and then you put your slide into the instrument. So basically it looks like this. You have an adapter. Then you need to teach the software, like where is your like the region of interest. And I would say up to now the smallest resolution you can get is 5 to 10 micrometers. This is like, you can compare it to pictures. This is like the pixel size you can obtain. Nevertheless, if you want to have this size with this resolution, it will take you like 2 days of measurement. So basically it's depending on what region you want to image, and then you decide what is the best resolution, and like you defining the size of the laser basically. And then when you teach the instrument where to look at, you get for each single shot a mass spectra, and then later on you can, for example, click on a specific mass and it will color code it. You, for example, show it in red, and then when you click on this, it's like distributed like that, and then you can merge it together and look what is present in your data. When I did my postdoc in San Diego, I just did it with this software, and it was horrible and tedious, and it was not really much fun because it took so much time to finally find something interesting. But luckily, there is now Skills, which is a statistical analysis software, which helps you to get quicker to the data of interest. So basically what you can do is you can co-register other images. So I will talk about shortly about this adipose tissue project where we had UCP immunohistostainings. We had the picture of the slice, we had the MIDI data, and then you can merge all the images on top to create like a really coherent figure what's going on in your tissue. And then the processing basically contains of denoising, denormalization, and then you can start statistical analysis. And for example, here's again a potato. So when you do this hierarchical clustering, it's basically a segmentation analysis. You see that like the outside where we have like the carboxymethylcellulose is different from the skin, is different from the inner part of the potato. And then you can look for, okay, what are those metabolites which are located at what site? And for example, in potatoes, the saponins are located on the outside, and that's the reason why you peel your potato before cooking it. And then you can say, okay, I want now to find the differences. So you can do co-localization of those biomarkers. You can do a rock analysis to really show that this is an interesting biomarker for a specific metabolite. And then lastly, I just want to show you the project we did together with Sebastian Diekmann. So the group of Professor Kleigrewe from the Chair of Molecular Nutritional Medicine, they are interested in brown and white adipose tissue. And the question was if, like, where are the cardiolipins located within this white tissue? So if you just take the tissue and you homogenate it and put it into the mass spec, you lose the spatial resolution of it. So in this case, we had the first slice was going to the MALDI, the second one he did UCP1 immunihistostaining, and then the third slice went to the LCMS because our MALDI unfortunately was a low resolution mass spec, and you nearly need to validate afterwards when you have identified markers what they are. So that's why we had like 3 workflows, and then we could show that cardiolipins are like basically located together with the UCP1, but when we use tissue from a UCP1 knockout mouse, we still could detect the cardiolipins. So with that, we got the conclusion based on the spatial analysis that this UCP1, the uncoupling protein, is independent of the occurrence of the cardiolipins. So this is published. If you want to have more information, feel free to read it. So which instruments can be used? So we used to have the Ultraflex 3. It is a good mass spec. Nevertheless, the newer ones have better resolution. And if you work with mass spec data and you have like, you can basically conclude from the M/Z ratio, the sum formula, it's the possibilities of metabolites or lipids are much less compared when you have like only 1 digit. Then it's like a little bit, you get some leads and you need to double check if it's correct. And this laser is less efficient compared to the ones which are out there today. So this is not applicable at the moment, but we have as the Klinikum rechts der Isar a timsTOF with the MALDI2. So here you have high resolution data, and you have this MALDI2 has extra more efficient ionization. This was developed by a friend of mine back in Münster. So basically they shoot with a MALDI and then they shoot into the cloud again with another laser, and with that they enhance the ionization efficiency. So with that, I'm at the end how MALDI imaging has been done here at BayBioMS. So we have the benefits that we can get the spatial distribution. It works for small metabolites. It's not working that great. Lipids are working well. Peptides and proteins are also possible, but it's still a lot of work to develop those methods. You have to do drug dissections on the slides, and then you most likely see the most abundant proteins. But there are groups out there who are doing this kind of imaging. And as I said, each step needs to be optimized for your matrix. And with that, you can create easy, fancy figures. But to make sense out of it later on, this is the next challenge. So how MALDI imaging is possible at TUM? So the BayBioMS at Klinikum rechts der Isar MRI has a RapidFlex, so this is going into tumor tissue typing, and they also have the timsTOF Flex here. We still have the image prep system here, so I think that it might be possible to get access to that. And then we have a new professor here, Strittmatter, and we'll also give a talk today about DESI. DESI is a different way of ionization, which I did not cover today. So this is like electrospray, what you guys know from our normal mass specs, and you don't need to do the step of adding matrix. So it's a little bit more simple, but I think the setting up the DESI source is more complicated and even more hands-on if you can buy the DESI source.
The first week is time, then if it works, it's fine.
So she will give more insights. So with that, I will.
Oh, thank you very much. I'm Nicole. I am in Professorship. We've got a very nice new logo, which one of Dania's colleagues actually made for us. And I'm going to talk to you today about mass spectrometry imaging and its application in pharmaceutical R&D. This is not necessarily what I'm doing now. It's what I've been doing in the past years before I came here. as my research group is still very new. I just started last October. My students started in April, June. We're not actually having that much stuff yet that I could show you loads of. Well, I could, but it's a bit fragmented still at the moment. Nothing is finished yet. Nothing has a story yet. So I thought it makes more sense to keep the talk about this time at AstraZeneca. So the drug discovery pipeline is really inefficient. So there is loads of different steps involved in this process, from finding a target to finding a right compound, then optimizing that compound, and doing the preclinical studies, and then doing phase 1, phase 2 clinical trials. And at every single one of those steps, you lose candidates. And that results in a huge amount of what is called attrition. So it's, yeah, all that money is spent without getting anything for it in the end because you don't have a product. And the overall success rate in the industry is something like 1.8% of the drug candidates that make it to the market. And most of those reasons why the drugs fail are shown here in this bar chart on the right. So you'll have the main reason is pharmacokinetics are not right, animal toxicity, so that would, yeah, if something's toxic in the animal, it doesn't even make it to the human. You've got also lack of efficacy as one of the major reasons. So we always joke that we can cure any disease in a mouse, but translating that whole thing then to the human is a whole other question. And many of those drugs just do not translate, unfortunately. And yeah, one of the big things that get iterated again and again within the drug development process is drug metabolism pharmacokinetics optimization. At AstraZeneca, they actually had, Owen just mentioned that because it really helped them decrease their attrition, and they had the 5R framework. So the 5Rs were, you have to identify the right target first. It has to be an efficient target. It has to be something that actually does something and that's translatable. You have to make sure that your drug makes it to the right tissue where it is needed, for instance, a tumor. So there's no point if it moves around in the blood if it doesn't ever make it to the tumor in a critical concentration. You have to ensure the right safety. So your drug obviously shouldn't be toxic or minimally toxic. Tolerable side effects, or a cytostatic drug, for instance, will never be non-toxic, but yeah, it should be tolerable. You have to select the right patients. So this is exactly especially going in the direction of now choosing, like stratifying your cancer. So you're not just throwing a drug on a cancer anymore, but you're really looking at the genetic makeup of your cancer. Where is the mutation here? And then have a drug that is specifically designed to target a cancer with that mutation. And that it decreases the market because you cannot just give it to any cancer anymore. But on the other hand, it increases your success rate with treating that patient. So that's where some of the really good results with like immunotherapy and so on come from. But then again, they don't really know why they work for some of those people yet. So really finding the right patients is a big area of active research at the moment. And you have to define the right commercial value as well. So this is a big reason why antibiotics are not pursued anymore. And also AstraZeneca shelved neuroscience, for instance, or many of the other things which just don't sell. So what AstraZeneca did in the end was cancer and cardiovascular diseases and things like COPD, so things that Western diet and our Western lifestyle makes increasingly common. Those are the kind of things we worked on a lot. So back to DMPK. So in DMPK studies, we're looking at drug metabolism. So how is the drug altered within the body? Pharmacokinetics. So how does the drug move around in the body? What is the time frames, which organs does it go to, and so on. And very important as well is pharmacodynamics. So does my drug actually result in a response? And this all feeds into so-called APME studies. So it's absorption, distribution, metabolism, and expression. And this is done for a whole lot of different advanced compounds. So that is often done at a point when they've got like a structure already. like a structural frame, but they're still optimizing some of the ligands on that frame. And then they have this iterative rounds of ADME studies to incrementally improve the compounds until they cannot improve it anymore. And yeah, this obviously improving it to the point where it's as good as it can be is also an important step towards reducing the attrition. Traditionally, the methods to study drug distribution are using plasma or tissues as surrogates, or plasma as surrogates and tissue homogeneous to look at the drug amount in there. Those are all done using LC-MS methods generally. Or you can have, once your drug is a bit further down the pipeline, they would start synthesizing radio-labeled compounds. So this is not done for every compound, obviously, because it's expensive and lots of work. But once, again, you settle on a certain type of compound, they would then start to synthesize those radio-labeled compounds as well. But there are complications here. So obviously, plasma is only a surrogate. It's not actually the tissue you want to look at. You don't know what happens in the tumor. You don't know what happens in the brain, and generally how it distributes in the body. You just know how long it circulates in the plasma. With tissue homogenates, you can see how it goes into your tissues, but you lose all that spatial information, like Karin explained already. You can also have contamination through blood. For instance, if you've got liver or so, or brain, you can still not really say whether the drug you're measuring now is because of the tissue or the blood. So having that spatial information here again helps, because you can, for instance, overlay your drug signal with the signal of the blood. So MSI is really helping a lot with those problems. With the radio-labeled compounds I've mentioned, they are not available early on in the pipeline. And they're quite costly as well. So why do we do MSI AstraZeneca? Well, ultimately, again, everything in industry is about reducing costs. So we want to reduce attrition through better understanding of the disease in our compounds. And MSI is great because it gives us untargeted and label-free investigations into how compounds distribute in a variety of different samples. So Karin just said they've done potatoes and carrots, we can do tissues, we can do all kinds of things. It's in situ mass spectrometry, so we're actually looking at things in the place where they originate from. So we're not looking at cells in culture, for instance, where they don't have the interaction with other cell types around, but it's just looking at everything in its native place, full complexity, everything's still there. And we can do multiplex analysis as well. So it's not just a single compound, we can do thousands of different compounds, and we can also in one run look at several different tissues as well. And one example to the whole body autoradiography is shown here. It's not really showing very nicely, but anyways. So those images are very pretty, very nice. It's of a drug that was radio labeled, and you can see the distribution here. So some white spots here, some white spots here. But the problem with those radio labels is that you cannot tell whether you're actually looking at your drug or you're looking at a metabolite. As long as that label is in there, everything will look the same. So, and yeah, once the label is gone, you don't see it anymore. So there's a big question mark what you're actually looking at here. And with mass spec, they've got two different masks, so we can distinguish them. So you can now actually see that this distribution, which we see up here, is made up of two different compounds, the parent compound as well as a dimethyl metabolite. Because in MSR we've got all of our analytes in the exact same spectrum, it's generally said that MSI is very insensitive, but it's not actually necessarily true. So if you've got cases like here, when you've got only very small areas where you've got drug localized, if you imagine taking a thing and meshing it up, it's like a ratio of 1 to 100 or so of having drug versus having no drug. This dilutes it possibly so much that you cannot see it anymore, even with LC-MS. But because we've got all of that little drug in one single spot, we can actually see it with mass spec. And that's what happened here. There was no successful detection with an LC-MS method, but we could see it quite well with not quite well, a bit strong, but we could see it with the MSI. So those here are noise peaks, the point here, so that indicates that it's not entirely high, but we could see it. And we've had that a lot in AstraZeneca as well, because actually the LCMS teams, they also run so many studies. Like they were 2 people and they were running 300 studies a year. They have no time to optimize. So they've got one method and they run everything over that. And this method actually was not sensitive at all. We were much more sensitive with the MSI. But yeah, we had less throughput. We had more time to work on things as well. Another thing is that we've got everything in one spectrum again. So generally it's that MSI is a qualitative method. So even if you've got 2 different tissue types on one section, like stroma and connective tissue, it's the same, tumor and connective tissue, you cannot really say this is 3 times as high as in this one without doing some degree of quantitation. They've got a different chemical background in those 2 different tissue types, so you cannot really compare directly how high your compound is in those 2 different tissue types. But there are ways to get around this, and one of the best ways considered is are those tissue mimetics. So we've worked a lot with that at AstraZeneca as well. Advantage of those mimetics is that you've got your compound distributed within the whole layer of your section. Because if you spot it on top, you again don't really know, does it ionize any easier because it sits on the top, or is it the same? So you don't really know. And there are some quite elegant, complicated but elegant ways to do those mimetic arrays these days. So in pharmaceutical industry, who's sending MSI groups? So generally it's only the really big companies. They've got AstraZeneca here. In the UK, they've got one. Boehringer Ingelheim is one in Frankfurt, actually. So that's probably the closest. Novartis has one in Basel, Genentech in the US, and GSK in the UK as well. And they actually also have a US one. For smaller companies, you've got the option of either collaborating with academia, which many people do, or you could go to CROs like Biotech, for instance. So what did we use MSI for? The most obvious one is drug delivery. So answering questions like, does the drug reach my target? And why is the drug where it is? So for instance, sequestering in 2 different tissue types or so. The second one is efficacy. So most drugs target a specific enzymatic pathway. So when we give that inhibitor, do we see the expected changes, like substrate going up, product going down? The third one is safety applications. So this is around questions like, does the drug do anything it is not supposed to do? We've had I'm going to show an example as well, like what are those crystals? And can we see early onset of adverse effects? So actually with MSI, we often see effects earlier than the histology can see them. Ischemia is a very good example, for instance. And the fourth one was model characterization. So this is a bit more esoteric questions, like what are the systems level differences associated with our models? So we've got a wild type and a KO model, and they want to see what are the changes happening within that model, and then how does that change again once we put the drug into the system. So just to give you examples of the type of things we worked at in AstraZeneca. So this is a very nice, elegant way, I think, of comparing different routes of delivery. So here we've got, for instance, in 3 different tumor models, we have deuterated free drug versus non-deuterated, so normal drug that got delivered by a nanoparticle. And you can see here in the exact same tissue section, the same imaging run, see how the nanoparticle delivery changes the distribution pattern of your drug. And the same is shown here on the right for a lung example, where they've compared, again, IV versus inhaled drug distribution. And you can see that intravenous, again, like here as well, it's very homogeneous, versus on the inhaled side, you've got a hotspot pattern. So this is taking this nanomedicine work a bit further. So once we can see how the nanomedicine distributes within the different tissue types, so we can then also tell how much drug makes it into the actual tumor compartment. So if you mesh your tumor up, you cannot really say that. Does the drug sit in the stroma? Does it sit in the tumor? You've got no clue. This way we can now say that. And although initially it looked like the lung tumor had by far the highest abundance, so you can see that here, the blue bar is always the highest. Once we look at the actual tumor cell compartment only, this looks a lot more similar. And you can see up here that in the lung tumor, 90% of that drug just sits in the stroma and doesn't actually make it to the tumor cells. So the second example, the short one, is where the crystals, or what are the crystals? So here it was a tox study, and the pathologists have seen crystalline deposits in the lung, and they didn't really know what they were, so they sent it to us. We section it, put it on the mass spec, and can then say, oh look, it's got the same mass as the drug, likely it's crystalline drug deposits. So that is then something where they would think about administering the drug differently, so that they, I mean, you don't want crystals in your lungs, it's not right. And the side example is around GLS inhibition. So generally in tumors, because of many tumors are quite hypoxic, and then this conversion here from lactate into the TCA cycle gets regulated down, so this doesn't really happen anymore, and tumors instead use this reductive glutamine carboxylation pathway shown here on the right. And the very first step of that is the transport of glutamine from the extracellular space to the intracellular space and conversion to glutamate. And this is this is catalyzed by GLS, so glutaminase, which makes it a very promising drug target. I mean, if you can kill off their metabolism right at the first step, that's a great thing. So those are the results here when they've given that drug to a mouse and compared control versus those tissues here. So it actually looks quite promising. We've got the glutamate going up because it doesn't get no, we've got the, I'm getting confused here. Oh yeah, okay, yeah, so glutamine we start with. So we've got the glutamine going down because it doesn't get produced anymore, and we've got the glutamate going down, and we've got the glutamine going up because it doesn't get transformed into glutamate anymore. And yeah, the changes in aspartate, for instance, are a lot more subtle in here. But the problem with this is, I mean, this looks amazing. Let's give it to patients. The problem is that glutamate is also a major neurotransmitter in the brain. So the question was then, okay, is it safe to give this to a patient? Because if it messes around with our brain, this is again a safety concern, and we don't really want to progress a drug that is doing that. So they have then given the drug to mice again and looked at the changes of glutamate, aspartate, and glutamine in brain, and we do see the same changes. So that was the point where that drug would have been axed, which is frustrating because sometimes you spend years working on a drug and then one day somebody comes and say, oh, did you know they axed that drug? Right, thank you for telling me. So anyways, one reason why I'm here now, because pharma can be a bit discouraging and you don't really know what you're doing things for at times, and yeah, I know it a bit more clearly. So this is my group on the left. I've got 2 PhD students, a postdoc, and a master student at the minute. And I'm going to give another talk at the SSD 924 early in November, actually. So for those of you guys who are a bit more into food applications, I'm going to try recording garlic and a few food stuff this week to tailor it a bit more to the audience. So thank you very much.